Well, first, thanks to the organizers of this. What a great event it's been so far. Everyone's had some really inspirational things to say, and I'm looking forward to hearing from Michael Eisen next. But for now, as Martin talked about, and if you don't mind, I'm going to just present on this one, so that I'm kind of tall, right? So I want to talk today about this new idea, data resuscitation. We'll come back to that in a minute. I'm not sure on the name. If you guys have a good name for it, we'll talk about that more in a minute. Let's see if that works. It does. Okay. So the reason I think that Martin and the organizers asked me to talk today is because I'm a little bit active in the hashtag SciComm these days. So that includes writing blogs mainly. So I blog on Medium, and I recommend that for anyone who kind of doesn't want to do all the time blogging, set up your own website and maintain all that, but rather you can write a little one-off blog article, very easy on Medium. And it's a great format just to write in as well. I also tweet on Twitter, Science Thomas. And also, I like to just talk about science. People who know me, my friends, usually can't get me to stop talking about science. So those are kind of that's probably why they asked me to talk. But during my time blogging, I've written about a lot of different things, generally for a lay audience. That's what I normally blog about. Recently I wrote about unstoppable killers here, the rise of antibiotic-resistant bacteria. I've written about economic impacts of science, and written about why the cold can't give you a cold, but it can make you more likely to catch a cold, and stuff like that. But today, when I realized that the group I was presenting in is a bunch of scientists, instead of just saying, hey guys, write blogs or something, I wanted to talk to you about an idea I had for my next blog, which I'm working on now, okay? And because I think this is something that could maybe be helpful for you guys, so I hope it's helpful for you. Another thing is it's something you may already know, right? But maybe just putting a name on it and thinking about it will help us be better at this. Okay, so this is something I found on the internet. I don't really know where to attribute it to, but it would have made a great PhD comic, right? I don't think it's from there. It's the life of a project, and I think any of you guys who do science, which is everyone here, are familiar with this curve, right? So you start out with this, best idea ever, the PI, right? 5 years of this, it's going to be great. Okay, science, nature, Nobel, But as you go on, you find, this is harder than I thought. Maybe it's not even working, and it sucks, and it's boring. And then down here at the bottom, this is Dark Night of the Soul, right? So, okay, that's like year 3 of grad school, right? Okay, but then what you do is you start to realize, okay, well, it'll be good, I'll finish something, right? And at the end you go, it's done, and it sucks, but it's not that bad, right? And I got that publication. Yeah, the impact factor is 1.5, but it's cool. Okay, so you all know about that. But what I want to talk about today is maybe this idea of how can we more effectively get from this dark night of the soul and get to save that project, guys. Get back to it. It's good. All right, so I want to just give you an example of something that happened to me, and in my PhD thesis, and it was about, right when I went to write it, that I realized that I'd missed a big thing. So let's talk about missing things. So first off, things don't always fit the model, okay? And in fact, I think I'm going to, at the end, kind of say that I think in the most exciting times, things don't fit the model. But this was not the most exciting times. This was just an average, kind of mediocre sort of thesis project. But and I'm not going to bore you guys with all the details here, but this is surface plasmon resonance, and we're kind of measuring the affinity of one thing for another thing. And the main point I want to make here is that the red curve is big, and the orange curve is in between, and the blue curve is little, right? Everyone's kind of good with that. Red is bigger than orange, is bigger than blue, okay? So that was one piece of data, and that is what kind of started out my project. We were going to compare those three. But then I did another experiment, reconstructing these receptors, now not in this very simple model, now putting them on a cell, using a virus, all this stuff, and suddenly now, in the context of being expressed on a cell, I had different results. Notice now that orange is bigger than red, Blue's still the littlest, okay? So what happened? I'm showing here, actually, this is figure 1 from this paper that we published. So in the end, obviously, this was useful in that it ended in a paper. So that's kind of the best place for things to end, right? But what did we do, though? This, my PhD took about 5 years, and the first 4 years of my PhD, after finding this disparate result from panel A to panel B, is I ignored orange, okay? Because this worked like this, and this worked like this, I said, it makes no sense. Let's look at things that make sense. So basically, this was what my whole PhD thesis focused on for 3 years. The red and the blue, okay? Red, we knew, was higher than blue in all of our experiments, so that made sense, right? But orange is the one that didn't make sense. Sometimes it was lower, sometimes it was higher, doesn't make sense. So we ignored it, okay? So we just stuck to this world. However, other people were also working on this, and they made a mouse, okay? And the mouse they made had the orange receptor, okay? And what was interesting is that these people found in their publications that this mouse had a really weird characteristic. That basically the B cells in the mouse, I was studying type 1 diabetes, and these receptors that I'm talking about, they're B cell receptors and they bind to insulin with different affinities. And what they found was that, in a mouse and in a human, insulin is expressed at really, really low levels. But this receptor really shouldn't bind to them because it wasn't supposed to have such a high affinity to bind in that low, in the endogenous concentrations. Yet, they were finding that it was binding insulin in this mouse. And so they published, some discussion basically saying, we don't understand why that's working. And what I realized when I was putting it all together and writing my thesis was that, and the paper that accompanied it, was that actually I had found that same observation here in, really biochemical methods and very, isolated, these examples that I showed before, where we had the, I was supposed to put this again. Right, I had basically already made these same observations. That when you express the protein as just a protein, you get red is higher than orange, and orange is kind of low, you're blue. But when you make it as a receptor on a cell, now orange is extremely, extremely strong. So I'd already basically made an observation that explained why this mouse had the function, or had the properties that it did. And so sure enough, I know this is a lot of words for a slide. You don't have to read them all. I put in bold the important parts. Orange here supports the development of diabetes in these mice, despite the fact that it had a lower affinity, as you would predict from the protein, because when you express it as a receptor on a cell, it has a much, much higher affinity, as I showed in that paper. Okay. But my point, though, was that for 3 years, I basically ignored, this receptor. And I really should have been paying attention to it the whole time. Because then I could have learned a lot more about this, thing that was expressed in a mouse, and, probably could have fit better with our understanding of diabetes. So in some ways, this is a cautionary tale. It's an idea of hindsight is 20/20, and how I'm trying to encourage you guys to look twice at your data, not ignore the outliers, stuff like that. And so this doesn't just happen to me, it happens to everyone. Here, in fact, is a paper called Making New Genetic Diagnoses with Old Data, Iterative Reanalysis and Reporting from Genome-Wide Data in 1,000 People. So this is actually a paper where the authors took their same data set that they published findings on in 2014, found about 27% diagnosis of genetic alleles causing disease in these children with developmental disorders. And they went and reanalyzed the data and came back with a 40% diagnostic rate. So they increased it by 13%, and they said that this would happen by systematically reanalyzing previously generated data in light of new knowledge. And that's kind of what I'm encouraging you guys to do. It was cool because Tim earlier talked about the repository of all the microarray data that's out there with over 2 million microarray data. And there's RNA-seq data out there all over, and a lot of people are doing these sort of meta studies right now. So you can, make a whole lab on something like this, but you can also just do this on your own data. You can go back to your own data, especially the stuff you thought that didn't make sense, especially the stuff that looks negative, and you can reanalyze it with a different, perspective, and hopefully in light of new knowledge, and maybe you'll find something that makes some sense. So, okay, cool. Hopefully I've now made a case that, at least for me, I see some things where when I went back and looked at it again, I was able to make some sense out of something that previously didn't make sense, and I've shown that also some other people have done the same sort of thing, and maybe you're thinking that you've done the same thing, or you're thinking, oh, gee, I should probably go back and take a look at that result that didn't make any sense last week. So I want to talk about maybe some techniques that we can all try to do, and this is kind of what I want to write the blog about, is this idea of how can we proactively work to, systematically or iteratively reanalyze things. Okay, so here are some of them. One is maybe spring cleaning. This is this idea that I'm throwing around where maybe, you all know the experiment, you do it and it's a big flop, right? And looks like nothing worked, or, oh my gosh, orange is way higher than red. What you want to do on this case is just, you want to just throw it away, right? Wow, I wasted that weekend working in the lab. I should have been out with my friends, and instead I get nothing for it, right? So you want to, I think it's some sort of PTSD thing. You don't want to think about it. So we just kind of throw it, suppress it, we walk on, we focus on the things that work and the things that make sense, or the to-do list. Okay, the victories. So that's fine. I think that's natural, and it's a good coping mechanism. So it's fine. But what I do think you should do is maybe, if you have a Mac, put a little flag on it. Make it orange or something on that folder that contains it. Because on a Mac later on you can look at that orange. I don't know how to do it on a PC. Maybe analog. You put it in a little notebook. Just the date. I don't know what the way is that you sort your files, but basically when you have that experiment that is really just, seems like junk at the moment, just put it in that little, book of shame or something, okay? The little date, okay? And then the idea with spring cleaning is that maybe once a year or at some periodic interval, you go back and you look at that, and you look at the stuff from a year ago, and you see, you just take a look again, Try to open it again. If I do a lot of flow cytometry. Don't even open that old flow cytometry file. Take the original ones, drop it into FlowJo again, start from scratch. Basically I'm saying don't open your same Excel, take the raw data, look at it again, okay? Spring cleaning. Hopefully you've made some new insights in that year. Maybe you've read a new paper or gone to a conference and things look different in light of the new knowledge that you have. Okay, so that's one idea. Another thing, when I've been talking to colleagues about this, is this idea of other people looking at it. Fresh eyes. Again, like I said, in science, we don't really like to talk about things that don't work, right? Although the majority of the stuff that we do doesn't work. I mean, at least for me. I don't know, maybe you guys are doing a lot better, but the majority of my time, I'm doing something and then in the end, it doesn't really work. But I don't, again, like to focus on that. I want to move on, focus on the stuff that does work. And so I'll often not share the stuff. People, in science have all made references to how, one day we'll start that journal of negative results, right? Everyone talks about it, yet no one wants to do it, right? It's a shame. Okay? But the point is we don't share negative results. We often don't think about them. You don't share it with your PI if you're a postdoc or a grad student. If you're a PI, you maybe don't share it with your colleagues or your, friends because you want to focus on the stuff that works. However, what I'm trying to encourage you to do is actually share something. Say, this didn't make sense. It really should have worked, right? Take it to your PI, the experiment that seemed like it didn't work. Show it in lab meeting, okay? That's one of the things I want to talk about too. Lab meeting is a great time to talk about this stuff because this is a connected thing to Fresh Eyes, diverse perspectives, right? Because the whole point is processing the data in light of different knowledge. It can be your own knowledge a year later, now it's new and different and you've changed your perspective. It can be a different colleague who has a completely different perspective. And in the end, by looking at your results from all of these different angles, you're more likely to find the truth. So those are kind of maybe it's two main concepts that I have: the spring cleaning, the fresh eyes, the diverse perspectives. But again, bringing it back to what these are all about, what I hope this kind of technique will do, it's about taking us from that dark night of the soul, right? It's about you've done a lot of work on this. You've got these results. They make no sense. They seem like you've wasted your time. But maybe you haven't, right? Maybe what's happened with the negative results is that simply in the framework that you're operating in, with the knowledge that you know at that time when you saw it, you just weren't ready to see the new idea, right? Because we understand the concepts only in the framework that we are, right? And I mean, I know there are people in this audience from a lot of different scientific backgrounds, but we talked about the scientific method earlier, and the scientific method is great. But the scientific method is also very strict, right? We're trying to make a prediction, we're trying to eliminate it down to one variable, we're trying to look at things, in a very clear framework. And in some ways, it's limiting our view, right? I mean, I'm not standing up here to decry the scientific method. But what I'm actually thinking about is things like machine learning and things like where we just toss a computer in there and say, have fun, see what comes out, What I'm trying to say is that if we look at things from just a strict angle where we have A and B, and hypothesis will give us result A or result B, and none of us want to talk about alternative outcomes, right? We don't want to say, well, what happens if we get nothing? What if we get C, right? The idea is that maybe C is the thing that overturns all of the dogma and the paradigms that we're working in. Maybe A and B aren't at all real, even though we designed the experiment to test if it's A or B. So by broadening our perspective, either through this idea of looking again at the things you think didn't work, or having other people look and encouraging and really using those diverse perspectives that other people have, maybe they'll be able to help you out. Maybe they'll be able to say, hey, what if it's C? And through that, hopefully then, help you go from dark night of the soul to paradigm shifting paper. So with that, thank you, and take any questions.